All right, good afternoon and welcome to the Immune Deficiency Foundation's SCID Compass Lunch and Learn. Today, we will be hearing a gene therapy update for ADA-SCID from Dr. Kohn at UCLA. My name is Emma Mertens, and I'm the Program Manager of Community Relations at IDF. On behalf of all of us at the Immune Deficiency Foundation, I'd like to thank you for joining us today for this program. We are excited to host this event for the IDF community. IDF is dedicated to fostering a community empowered by education. We want you to remember that IDF is committed to our community, serving you as a trusted resource through the use of technology and innovation. We are here to give you the tools and information to become empowered and offer you our compassion, understanding, and support to emphasize that you are not alone on your journey. Today's program is part of IDF's regular series of bite-sized programming that will provide diagnosis-specific information and support to our community wherever they may be. Today's program is supported by Chiesi Global Rare Diseases, and we will be hearing from them later on today in the presentation. Before we begin, I would like to point out a few housekeeping items to keep in mind for today's program. This afternoon, we are using the Zoom webinar feature, which means that attendees should be able to see the slides and our panelists and also hear myself and our panelists speak. Attendees will not be able to activate their video camera or their microphone. There will be the opportunity for questions at the end of the presentation, and you are welcome to submit any questions you have in the control panel box at the bottom of your screen where it says Q&A. Please do not include any personal health information, as all questions will be anonymous and read aloud. Oops. A brief disclaimer. Please remember the information presented during this forum is not intended to be a substitute for medical advice, diagnosis, or treatment. We are here today as a trusted source and friend to provide you with information. Always seek the advice of your physician or other qualified health provider with questions concerning a medical condition. Never disregard professional medical advice or delay seeking it based on information presented during an educational forum or event. Many of you tuning in today are doing so because you are an individual, parent, caregiver, or friend to someone living with severe combined immunodeficiency or SCID. SCID Compass, an educational program of the Immune Deficiency Foundation, was created in 2018 with the purpose of guiding parents, families, and the medical community through the journey of learning about this rare and life-threatening medical disorder and find support to navigate the health challenges along the way. All of our SCID Compass resources can be found by visiting www.scidcompass.org. The website offers a robust variety of online and printable materials for anyone eager to learn more about this condition or share information with others. Topics cover every step in a family's SCID journey, from newborn screening to return home after treatment, and everything in between. You can view the language in Spanish, English, French, German, Portuguese, or Tagalog by clicking on Select a Language in the upper navigation. We also offer monthly lunch and learns and support all of the great programming that IDF offers to the PI community. This fall, we hope you will join us for IDF's largest event of the year, our 2022 Primary Immunodeficiency Conference, taking place October 6th through the 8th. Registration is now open, and you can do so by visiting www.primaryimmune.org/conference. And now I am so pleased to introduce our presenter for today. Donald Kohn is a distinguished professor in the departments of microbiology, immunology, and molecular genetics, pediatric hematology and oncology, and molecular and medical pharmacology at the University of California, Los Angeles. Welcome, Dr. Kohn.
Okay, thank you very much, Emma. The distinguished always makes me smile. It's a university title. Anyways, I saw many friends on the meeting. And so I thought for today, since I think many of you know sort of the fundamentals of gene therapy, I'd sort of start a little further into the talk. I've submitted the slides to IDF as a PDF to post. So there's the first 20 or so slides are kind of a background on gene therapy. But I think what, the interest for today is to where are we with our trial? And so I want to get there as quickly as I can. So I'll start here, just a little background on ADA-SCID or adenosine deaminase. We know from now more and more data, it's the cause of about 10 to 15% of cases of SCID. And we estimate there's about 10 babies born with ADA-SCID per year across the US and Canada, although with such a rare disease that it varies. And of the 20 or so different genetic forms of SCID that we know, it's the first one where we understood it to some extent, the biochemistry, the genetic cause, and it was the first one where the normal gene was isolated or cloned, and that allowed gene therapy to be considered because now we had a normal copy of the ADA gene. We could think about putting it back into patient cells. Sorry. And so what we know is that ADA deficient patients have profound lymphopenia. They're missing all three types of the major lymphocytes, T, B, and NK cells. And we know the biochemistry is from toxic metabolites that build up in the absence of the enzyme. And the treatment options for patients are an allogeneic or a transplant from a donor. That could be a matched sibling donor if one is available. In the absence of that, either it could be a matched unrelated donor, or usually a non-family member that shares the major tissue types, or a half-matched transplant from a parent. There is, as we know, an enzyme replacement therapy that was first approved in 1990, which is adenosine deaminase enzyme conjugated to PEG, which prolongs its survival in the serum. And so twice weekly injections of PEG-ADA can detoxify the metabolites and allow the immune system to recover. And then there is autologous transplant with gene therapy that I'll talk about today. And so just to set the background, this is a slide from a paper showing the outcomes for allogeneic transplant, or transplants from donors for ADA-SCID patients. And this is a paper that was published a decade ago and really reflects patients from 1, 2, and 3 decades before that. So this is old data, but this shows the survival of patients with SCID, with ADA-SCID specifically. And remember, this was before there was newborn screening. So many of these patients didn't get diagnosed until they already had a severe infection, which greatly increases the risk at the time of transplant. But so in this sort of classical paper, there was about 90% survival for SCID patients who had a matched sibling. But if they had an unrelated donor, it was only about 70%, and even lower with a mismatched unrelated or a haploidentical donor. And so when gene therapy started, it excluded patients that had a matched sibling donor because they had quite good results. But for the patients that need an alternative donor, gene therapy could be potentially a better option. Fortunately, I want to make sure that everyone in the ADA-SCID community is aware of this paper that I'm very proud of, that came out of the Primary Immune Deficiency Treatment Consortium that I'm a part of. And as many of you may know, this is basically a multi-center organization of transplant centers across the US and Canada, 47 sites that collect data from patients with SCID, WSCOT, CGD, and some other disorders, and primarily do studies on the results. So we just did a paper that took us about the last 3-4 years to do, looking at all the ADA-SCID patients who had been entered into the PIDTC registry. And there were 131 patients going back to 1982, and we made our data cut up to 2017, so that we had at least 2 years follow-up from the patients transplanted in 2017. And this is a graph like the one I showed you before of overall survival. And so patients who either went right to transplant or got enzyme therapy before they had transplant had about a 70% survival, and this is all donors. Whereas gene therapy really looked quite good. All the patients who got gene therapy survived. Many of them all of them, in fact, were first stabilized on enzyme, and many of the gene therapy trials, in fact, excluded you if you had an infection. So there's some patient selection that makes it look good. But what's the really important message we found was, if we look at just the more recent last 2 decades, a decade and a half, for the outcomes so patients transplanted since 2000, those that got a transplant, whether they got enzyme or not before, were grouped together, had about a 90% survival rate, and it was statistically not significantly different than enzyme therapy. So as a transplanter, we always say we're getting better, but I think there's really evidence that for all SCID patients having some type of transplant, the outcomes are really quite good these days. And even in the 2000, probably the first decade was before newborn screening. So if we looked in the most recent decade, they might even be better. But the idea of gene therapy is that instead of using cells from a donor, the best available match, is to use the patient as the donor and take their own cells and correct them. And so I've been involved in this since I was a fellow with Mike Blaze in the mid-'80s. And this is sort of our, at UCLA and CHLA before that, our lifetime treatment of ADA-SCID patients. We've treated somewhere in the mid-50s total in a series of trials. So back in 1993, we treated 3 patients using cord blood with sort of version 1.0 of the growth factors and vectors, and got very little engraftment from it. We spent a decade in the lab basically trying to make how we process the stem cells better and better vectors. And so then we opened up an IND with FDA in 2000 using a retroviral vector we had made and the better growth factor conditions we had found through the 90s. And the first 4 patients were treated without any chemotherapies, just taking their bone marrow, putting in the vector and giving it back to them. And then, as I told you, as I'll show you, we then started giving chemotherapy. And so in the middle of that, we were on hold for 3 years after there were leukemias that developed in some ADA-SCID patients in Europe. And it took the FDA about 2 years to work through the risk and allow us to reopen. And during that time, a paper was published from TGET Institute in Milan, treating 2 ADA-SCID patients with relatively similar cell processing vector, but they gave them conditioning chemotherapy. So that's some medicines before the transplant to get rid of some of the remaining bone marrow and make space for the cells. And both of the patients they reported had a nice engraftment. So our last year of hold was us modifying our protocol to add the same kind of busulfan chemotherapy that they had. And so since that time, every patient we treated has gotten lower dose of busulfan as conditioning. And so we treated the remaining 6 on that trial. Then we actually moved cross town from CHLA to UCLA, which are different institutions, all confusingly named similarly. And we opened up a phase 2 trial and treated another 10 patients with a retroviral vector. But during that time, it was becoming clear that lentiviral vectors were coming out as a better type of virus. And so we then moved from a retro to a lenti under a new IND with FDA using this vector that I'll tell you about. And so I'm going to talk about the trials that we've done with that lentiviral vector. And the reason for lentiviral vectors is that, as I mentioned, in the early trials for X-linked SCID at least, several of the patients got leukemia probably from the virus. So the early vectors that were made from mouse viruses have a very strong element called an enhancer that drives expression of the gene. So you made a lot of the product like ADA, but they can turn on a nearby gene. And so when we treat a patient's cells, we might treat 10, 50 million cells. The virus goes into different places in the chromosome in each cell. And if they happen to land next to a cell's growth gene, and the enhancer turns that gene on, that can start triggering that process to leukemia. And so sort of in the mid-2000s, with that problem having occurred clinically, so a second generation of vectors were made in which those enhancers are deleted. We call them self-inactivating or SYN vectors. And so these newer generation vectors use an internal promoter to drive production of the messenger RNA from the gene, but they don't have enhancer activities to turn on a nearby gene. And so this is from London, Bobby Gaspar and Adrian Thrasher. We were at a gene therapy meeting, I think in 2008, and we said, let's work. They were doing trials with retroviral vectors also. So we said, let's work together to make a lentiviral vector and do it in protocols together. And so this is the vector, and it's basically that type of SYN virus with a small promoter to drive expression of the ADA gene. There's various tricks where we can codon optimize and we get more bang for the buck. So this vector actually makes more ADA per copy than the mouse virus we had used before. And so between Great Ormond Street Hospital in London and our hospital at UCLA and a few patients treated at the NIH, we've treated more than 50 patients with this lentiviral vector. And this was just sort of our pathway to get to the trial, just to show you sort of what it takes to do this from academia, where we have limited resources that are dependent on external funding. So we had a program project grant from the National Heart, Lung, and Blood Institute that funded multiple projects. This was still at CHLA. And under that, we did the work to test the new vector and bring it to clinic. We then went through the first committee review, which was the NIH RAC, which actually no longer exists, but they used to review all the protocols. We then applied for funding from a gene therapy resource program that the Heart, Lung, and Blood Institute had that paid for the vector to do our screening studies and the toxicology study. We had a discussion with FDA. We got that grant. They made the vector. We got approval through our IRBs. Then we applied for the grant to do the trial. And this was a U01, which is a grant in the order of $2-3 million that would pay for treating 10 patients in the trial we had developed. And just a shout out to Linda Griffith, who many of you know, who's the program officer of this grant and really made everything that followed possible. It then went through the IRBs of the NIH where Fabio was. We did all the preclinical studies. And so it took almost 4 years to get from, hey, we should make a lentiviral vector to moving into trial. And in fact, we were delayed another year. So we started treating our first patient in 2013. And then this was the schema that we followed at that point. So we would be referred to a patient with ADA-SCID. We'd go through a series of calls with the parents. Usually they were not in the LA area, talk about the protocol, talk about options. And then on a separate call, if they wanted to go forward, we would have them sign the consent. The baby would have their screening test done at their home hospital, so they wouldn't travel until we knew they were eligible. And that was things like a chest x-ray, an EKG, some blood tests. And so if they were eligible, they would then come to LA, would actually repeat the consent face-to-face. And on Monday, they'd get admitted to the hospital. Tuesday morning, they'd go to the operating room and we'd harvest their bone marrow. So we'd do a bone marrow collection from them, and a PICC line would be placed, a long IV. Then the bone marrow would go to our laboratory, and it would take 2 days to process it, to isolate the stem cells and add the ADA gene with the vector. If by Tuesday night we knew we had the right number of cells to proceed, the patient would get a single dose of busulfan, again, to make space in their marrow. And then that gave about a day and a half of the busulfan to leave the body. So on Thursday, we'd take the cells from the laboratory over to the hospital and inject them through the patient's PICC line. And that was the treatment. And if all that went well, at a month out, we would stop their enzyme replacement and then follow them for, this trial was 2 years of follow-up with sort of monthly and then every 3-month visits to check how things were going. And so we treated the first patient under this trial in August of 2013. She's 9 years old now and doing well. So each one of these rows is a patient showing the time that they had transplant and their 2 years of follow-up. And when we finished that, we had many more patients referred to us. We still had money left in the grant. We still had vector. So we actually treated another 10 patients. So we treated a total of 20 patients plus one actually patient off trial who was too sick to qualify for the trial but needed it. And so between 2013 and 2018, we treated and followed out to 2 years this group of patients who got the fresh cell product. But we recognized at that time that this model of having the patients come to one center and get the cells taken across the street from the laboratory to the hospital isn't a practical model, and that we were, for other trials and other people, were getting to freeze the cells at the end of the processing, and then you can give them back at a later time or ship them from a central manufacturing plant to a clinical site. And so we planned a trial. And then as we were working on that, a company was formed, Orchard Therapeutics, and it licensed this gene therapy from our university and from University College London. And so they helped us develop the protocol, and we received funding from the California Institute for Regenerative Medicine to do what we called the frozen trial. There's the frozen, where we would test how well do frozen cells work compared to the patients who'd been treated before with the fresh cells. And so it was now split into 2 admissions. The first one was just to collect the stem cells. So after the consent and screening, the patient would come, we'd harvest their bone marrow, and they would go home back to their home house the next day. We would do the same identical gene transfer, but at the end of it, we would freeze the cells. And there are ways to freeze cells that they remain alive, and you can thaw them out and give them. And that allowed us like a month or 2 to do all the testing and make sure everything was right with the cells before we were giving them, before we had done all the testing. And this is certainly the way that most drugs are manufactured. You give them only after it's been tested. And so it takes about 5 weeks or so to do all the testing. So then the patient would come back for the second admission. And now, because we have the cells frozen, we can actually take our time giving the chemotherapy, and we split it into 2 doses, because it turns out there's a big person-to-person variability in how quickly they clear the drug. And so by giving 3/4 of the dose and measuring that patient's clearance, we could adjust the second dose. So they all got very close to the exact amount we wanted them to be exposed to. And then on Friday, the cells would be thawed at the bedside and infused through the patient's PICC line. And the same follow-up. So the goal here was to do everything exactly like the fresh trial, other than giving the cells that were frozen to see if they worked as well. And so for this trial, we already had a referral list of patients waiting. And so we went through all 10 patients in an 8-month period, which is, I think is pretty phenomenal from our lab group to do that. So red is when we made their product, and green is when they got the transplant. Then they were all followed for 2 years. And so the IND amendment was to use the frozen cells was accepted in December 16. It took us about 8 months to come up with all the data we needed to support the change to the frozen cells. And then we treated 2 more patients in 2018 with some of the remaining of the virus. And then it left our hands. So then it went to Orchard Therapeutics, and their goal was to take all of the data from these patients and get FDA approval to market the drug. And this is just a map of where the patients came. So we got very good at Canadian geography. So about 1/3 of the patients came from Canada, about 2/3 from the US, and 1 patient came from Lebanon. And this small group of people did all that work that I just showed you. So they did the cell processing. Dana Terrazas, who many of you who may know as the research nurse, worked up all the patients, brought them in for their transplants. And then the other group managed the data and the regulatory part. And so we published the results of our trials pooled with those from the London trial just last year. And so the trial was done primarily at UCLA's Mattel Children's Hospital, Great Ormond Street Hospital in London. And Dr. Claire Booth of Great Ormond Street Hospital was the lead of the trial for London. So we're co-first authors for the trial. And what we reported in the paper were 50 patients, 30 from the US, 20 from the UK, who were treated under a common protocol with the same vector. In fact, the batches of vector that were made were split 50-50 between us and them. So patients got the exact same vector. The cell processing protocols were very similar, and they got the same conditioning with the busulfan. And so when we looked at our outcomes from the trial for this paper, first of all, this is survival. And so the London actually started a year before us. So they had 3 years of follow-up on their patients in the green lines throughout these figures. We started later, so we had 2 years of all patients when we submitted the paper. And as you can see, this is a survival curve. 100% of the patients are surviving still. And then this is event-free survival. So event-free means surviving and not needing another transplant or go back on enzyme. So meaning, did it work? And so we each had 1 patient in whom it didn't work. 1 patient, the cells just did not engraft. We couldn't figure out why. She's gone on to have an unrelated donor transplant and is doing well. And then 1 patient in London did not mobilize very well, so got very few cells. He was quite sick and was on a lot of antibiotics that we think might have decreased the mobilization. So we got a very low cell dose. And at 1 year, they decided that he did not have sufficient engraftment and put him back on enzyme. So 2 out of 50, it did not work, but 48 out of 50 it did. So 96% success rate. And just a little bit more data from the paper, so we followed a lot in the patients at each of their visits when they came in. And this just shows their T cell counts. So when they came in, they were on enzyme and they had about 300 T cells, which was probably about 299 more than they had before going on enzyme. But then after the gene, so we stopped the enzyme at 1 month and counts dropped a bit. But then over the course of 1 to 2 years, they came up not to the fully normal range. For children about this age, they might have 2,000 to 3,000, but certainly this is sufficient to be protective. And none of these patients have had any SCID type infections. They've had normal childhood infections and dealt with them well. So it seems to be sufficient numbers of T cells. And their antibody levels also came up. So this is IgA and IgM followed from the beginning. IgG we didn't start following until patients got off their immunoglobulin replacement, but those that have been off have held their immunoglobulin levels fine. And so I think between the 50 patients or the 48 evaluable patients, I think 46 or 47 of them are off immunoglobulin. 1 or 2 did not get sufficient B cell recovery to get off immunoglobulin. And then we also scored just the infections after the transplant. And these are per year. And so this is like, these are very few. And so we had a few infections that were mainly line infections in the months after transplant, but beyond a year, very few. So each one of these, I think, is one patient getting admitted. Typically, they were like viral gastroenteritises, but because of concern that they had SCID, they were admitted to the hospital. But no serious infections, but they were hospitalized. They counted as severe. And then the last piece of data, this was sort of buried as supplemental figure 8, but this is from the US trial comparing the outcomes of the 20 who got fresh and the 10 that got frozen cells, showing that they're really indistinguishable for the copy number and the granulocytes, which is sort of the engraftment in mononuclear cells, the T cell counts, and red cell ADA activity. And all the parameters we looked at were not different whether they got fresh or frozen cells, which it would validate the use of the frozen cells, which, as I said, logistically, it's much better to use. And so, so just to review, so this protocol was developed jointly by University College London, Great Ormond Street Hospital, and also UCLA. We did the preclinical trials. I showed you the trial results. And just in terms of engraftment, 29 out of the 30 patients in the US and 19 out of 20 have sustained engraftment. The paper went to 2 or 3 years, but the patients are out as long as 9 years. And we've seen no decrease in immunity over that time. And so, academic medical centers, hopefully we pioneer new therapies, but we're not drug companies. And so for this to go from our hands, we can get a research grant to do these trials to make them commercially available. In the US, it needs to go to FDA for a biological license application to get approved for marketing. And so it was licensed, as I said before, from our 2 universities that own the intellectual property to Orchard Therapeutics in 2016. And so just to step back, this is sort of the timeline for where we've been. So Eloise Giblet identified ADA deficiency as a cause of SCID in 1972. The ADA normal gene was first isolated or cloned in about 83. First vectors were made. Then the NIH did the first trial targeting T cells from patients on enzyme therapy in 1990. Multiple trials were done using gamma retroviral vectors, first in Italy, but also in the UK and the US. And in fact, the Italian vector they developed, a gamma retroviral vector, was approved by the European Medicines Agency as Strimvelis. Lentiviral vector started in 2012 in England, and we are here now. And so then to tell you now where things stand since that point, as I'm sure many of you know, this past May, Orchard Therapeutics decided that they weren't going to further develop the ADA-SCID, and they announced they were going to return the license to the universities. They had really not made the progress they needed to be ready to submit it to the FDA, and sort of for financial reasons decided that they weren't able to make the investment it would need to get it across the finish line. And so we immediately began working to be able to start treating patients again under a compassionate protocol. And so we developed a new protocol for treating more patients. And we, in fact, got approval at our local IRB and our biosafety committees in October for the protocol. And so this is the title of it. So phase 1/2 trial, efficacy of cryopreserved autologous mobilized peripheral blood. So one of the main changes we're making is, instead of using bone marrow directly as the source, the use of mobilized peripheral blood, where the cells are chased from the marrow into the bloodstream and collected by leukapheresis, is really becoming much more the dominant mode, and especially in older patients like 5 years old and 6 years old that we now have from our early referrals. It's a more abundant source of cells. And so we propose to using that as the stem cell source as one of the sort of the novel scientific questions. And then it turns out there is a batch of the same lentiviral vector preparation from Indiana University that Orchard Therapeutics had made for them for the comparability studies, which has now come back to us and is sitting in our freezer in our manufacturing facility here. We plan to use mobilized peripheral blood stem cells, as I said. And then there were a couple of other small tweaks to the cell processing that have been, sort of come into the field since we last treated these patients, that allow us to use less vector per patient to treat more patients. And so our new protocol incorporated all of these changes. So of course, then we need to go back to the FDA and the IRBs to get approval. And so a little more of the chronology. So the IND, which is the FDA permission to treat patients, and to talk to the FDA, came back to us finally in January of this year. It took about 6 months just to untangle, and it is very complicated. And a lot of lawyers have spent a lot of time trying to work on the return of all the clinical data, everything else to us. And so the FDA IND came back to us in January, I think, 14th, and along with the pledge for the vector, which has come. And then most importantly, the grant that we had gotten in 2016 with Orchard, there was still money left in it. And CIRM, who's very, very in favor of this moving forward, allowed us to apply to again become the leads for the grant. And so the grant came back to us end of January with enough money to cover all the startup costs we have to get to the trial, and then to treat, we think about 3 to 6 patients, which really depends on how much the clinical costs will be covered by the grant versus by insurance or home hospitals. But as I said, we need to amend the IND with the FDA to get approval for these changes. And we're making changes, I talked about, both in the clinical and the manufacturing, the chemistry manufacturing controls as the cell manufacturing. And so once we had the standing as sponsor, having the IND back, we requested a meeting with FDA to talk about our plans. And that was scheduled 3 months from the time of the request, which is their typical waiting period. And so a month before the meeting, we sent them a briefing package that included the protocol that had gone through the IRB and all of our manufacturing plans, which was, 95%, 98% as we had done before. But to the FDA, I've learned similar means different. And if there's any difference, they want to know what it is and will it impact the product. And so we gave all that information to them a month in advance and had a meeting with them the end of April. And so at that meeting, the FDA requested a lot of changes. So it was a 1-hour meeting that was one of the most intense hours of my life, going through all this to make sure we understood what they were asking us. So they wanted us to change the objectives of the protocol. And I'll show you those in a minute. And so they recommended that we make the primary endpoint for the trial survival as something that's, black and white endpoint, and, certainly makes a difference to patients. And so we did that. They emphasized they want us to record infections as we're already doing. They also asked that we do neurodevelopmental testing. As many of you may know, patients with ADA-SCID have increased frequencies, although it's still in a minority, of neurologic problems like attention deficit disorder, as well as hearing defects. And so they thought it was important that we look at, does the therapy affect those? There's actually very little known about, are those fixable, or are they sort of present at birth and they don't change over time? But the FDA was quite insistent that we add this to the protocol. So that took some time, finding a neurodevelopmental psychologist who can do the testing, which we now have in place. And then for the drug product manufacturing, they want more data for how our vector performs using mobilized peripheral blood. We sent them, we thought, a fair amount of tests we had done with it, but they wanted some specific data. They wanted more information on the vector batch, and we have that for them. And then more about more information about the processing. And then one of the things that has taken about 3-4 months is they have now added, for when you give gene modified cells to a patient, they want you to measure how many copies of the vector are in, not every cell, but in representative number of cells. And we gave data to them on that test that we had developed in the April meeting, but they asked for a lot more data. And so we've probably done 2,000 colonies that we've measured the vector copy on to develop a complete data set. And so hopefully within the next week or two, we will be submitting all of this to them. We have about a 30-page memo, which is all of their comments and our responses. We have the modified protocol, we have the modified manufacturing plan. So if this goes back to them by the end of this month, they then have 30 days to review it. They can put us on hold if they think there are still some significant changes they want, or they can accept it. And once they've accepted it, we then have to circle back to our IRB, since the protocol they reviewed last October has been changed. And so we were hoping to be treating our first patients in August when we got the IND back in January. All of this extra work is probably adding 3 to 6 months, and our current working target is to enroll the first patient in January 2023. And we can discuss all this. I will be done pretty soon. So sorry, this is a pretty detailed slide. So these are the new objectives to the protocol. So we made the primary objective, as I said, overall survival. And I think the FDA looks at, this trial could be enough to get approval, depending on how it goes. And so they like to make approvals based on a really hard and fast endpoint that is quite meaningful to patients, obviously. And then we have secondary endpoints to look at safety and efficacy parts of the study. So we always look at safety. We record adverse events in the clinic. We make sure there's no live replication competent lentivirus that's never been seen. We make sure there's no vector-driven expansion of stem cell clones like occurred with the gamma retroviral vectors, which we haven't seen in the previous trials. We still think it's important to look at event-free survival because I think that defines failure or success of the treatment. And so we will be recording that. We have a whole detailed measurement of how we'll record infections and hospitalizations related to infections. And then, as I said, we developed a neurodevelopmental module. And so our plan is that at the time the patients come for their stem cell collection, they'll have a half a day of neurodevelopmental tests to record their behavior and their cognitive function. We'll also do a hearing test at that point. And then at the end of the study, at 2 years, we'll ask them to come back to LA to repeat those. And I think there's actually good value in that. There's very little in the literature to know, for example, today's population picked up by newborn screening, treated very early, what's their frequency of these kind of abnormalities, and does it change over 2 years after gene therapy? So this will be sort of a little study into itself that we are now ready to do. Then we'll also be recording when immunoglobulin replacement stopped. The FDA wanted us to have very precise criteria for when we stop it, since it's a success fail criteria. And so we've defined that very precisely of who gets to stop their immunoglobulin. Then we have exploratory objectives to look at how many cells have the gene, how many different vector integrants are there in the population, sort of biochemically, how much ADA enzyme are they making, have they cleared the bad metabolites, and then a whole battery of tests to look at their immune function, T cell, B cell, NK cells, immunoglobulins. And if they get off immunoglobulin, we'll then vaccinate them with their childhood vaccines and measure antibody production to those as a real good test of immune function. We'll also be assessing their performance status by just scoring, and then asking the parents to fill out a quality of life survey before treatment and then at 1 and 2 years. And so the protocol ultimately for the patient will be similar to as it was in the frozen trial, except we're using mobilized peripheral blood. So after the consenting and screening, the patients will come to UCLA to have their stem cells mobilized and collected. We'll then make the cell product and freeze it, release it, and then be ready to go. And the patients will come back, and the transplant will be just like it was in the prior frozen trial. And so a big topic of discussion, I assume, is the waiting list. And so when we stopped treating patients in 2017, we didn't really know we needed a waiting list. We thought Orchard would be up and going. Their goal was to be approved by 2020. But we kept getting referred patients, and kind of still, when there's a new ADA-SCID patient, often the doctor or the parents will write to us to ask about the availability of gene therapy. So we've kept the list since that time in the order of referral. And we think that the most equitable way to treat patients is to follow the order of this list. So this is the number of patients referred in each of those years for a total of 26. Now, we haven't kept up with all these patients, so it's possible that some of them may have had other therapies. They may have had transplants or other treatments. So we're not sure that we have 26, but we know the first 3 that we'll start with for sure. And so, as I said, we're hoping to get started in January and hopefully treat these first 3 patients in the first half of 2023. And so I'll just stop there by acknowledging this is my lab group, and they do sort of all the things I talked about. They make the vectors, we're doing gene editing, they do the clinical work. This is all of our support. We have support from many agencies, including the NIH, we have FDA for a retro trial. CIRM has been a major funder for all of our work in multiple diseases. And then several foundations, the Wiscott Aldrich Foundation, the Jeffrey Modell Foundation, SCID Agents for Life, are funding ongoing work in the lab to develop new treatments. And then we have many collaborators around the world for our various studies. And I will stop there so that we have time for discussion. And Emma, I don't know if you want to do the discussion now or if we want to hear from Dr. Trevino first.
Sure. So first of all, thank you so much, Dr. Kohn, for taking the time to provide us all with this important update and for sharing your expertise with us. I know everyone is probably really excited for Q&A. So just a reminder, if you would like to ask a question, please enter your question in the Q&A box at the bottom of your screen. And before we dive into Q&A, we are now going to hear from one of our sponsors at Chiesi. So I'd like to give a welcome to Andre Trevino, Associate Director for North America Patient Advocacy and Rare Diseases. Welcome, Andre.
Thank you, Emma. Thank you, Dr. Kohn. I'm not a doctor. I'm a rare dad. That's the best way I can describe myself. So hope is that thing inside us that insists, and what a perfect example of insistence we just heard. So I'll be quick because I know this is important. I'm Andres Trevino. I work for Chiesi Global Rare Diseases. First of all, thank you, Immune Deficiency Foundation, for the opportunity to sponsor this important Launch and Learn. And thank you, Dr. Kohn, for this important update. I deeply understand the importance of this because, as I told you, I'm a rare dad. My son, Andy, had NEMO, which is also a form of primary immune deficiency. We're very fortunate to receive care from Dr. Raif Jeha, from Dr. Jordan Orange, and Dr. Sung Yun Pai, who actually transplanted my son, who is now 23. He received cells from my daughter Sophia 6,530 days ago. And he's now 23 and doing well. So I just want to show you two quick things. And I'm going to share my screen here. And I'm going to make sure to share with sound. One thing is to thank SCID Angels, Heather and Barb, that actually helped us create this disease awareness website. If you haven't been there, please go to ADA-SCIDinfo.com and you will see Eliana, you will see Phoenix, Emily, you will see down here as well, Clementine, I love those cheeks. You will see also, of course, Aria, Damari. And if you haven't watched the story of Zena and Ray Ray, please do so. They are a beacon of hope. I've watched this story so many times and I always get goosebumps. If you haven't done that, please visit. And then the other thing I want to show you is a video from Giacomo Chiesi, who's the head of our Rare Disease Business Unit. And with that, we finish. It's a 3-minute video, and then we move to Q&A. So here we go.
Family businesses tend to focus a lot on the long term. We as a family and as a company, we look forward to the next generation of people and not necessarily the next month or the next quarterly report. So we have that long-term influence. We are very close to the patients, to the caregivers, and their families. Our mission in patient advocacy is let the patient voice shape who we are. I've recently met Vicky and Fred at the Jeffrey Modell Foundation. I've familiarized myself with the story of their son, who was born in 1970 and was diagnosed with a rare immune deficiency. Jeffrey said throughout his life, do something. He was right. We should all do something about rare diseases. If we think about rare diseases, they are a true significant societal unmet need, with an estimated 350 million people affected by a rare disease globally, 6,000 to 7,000 rare diseases, of which only 5% have an approved treatment or an experimental therapy today. Most rare diseases don't even have a good diagnostic device or even a protocol, and the average time to diagnosis is over 5 years. And only by putting the patients in the middle can we as a company support society in achieving its goal, health for all at all ages. The first one is a general lack of awareness. And there's also, we feel, a lack of shared responsibility by the different stakeholders. Society and the governments make decisions which are reasonably based on large data, not small data. But by doing that, society unfortunately leaves rare disease patients behind. COVID-19 has thrown a rock on everyone, and even more so on rare disease patients. In 2020, we launched Chiesi Global Rare Diseases, and a little more than a year, we've created an organization of 200 people with a presence in more than 20 countries, with a full integrated structure which is patient-centric and research and development driven. We're currently present in 3 different areas: inborn errors of metabolism, hemato-immunology, as well as ophthalmology. One of the reasons why we set up Chiesi Global Rare Diseases is to continuously put the patient in the middle of everything we do, as well as helping the rest of the company evolve and take its dedication to all of the patients that it currently serves to a whole new level. As a family business and as a benefit corporation, we are here to stay in rare diseases. We're committed for the long term.
Thank you again for the opportunity, Eddie. Back to you.
Thank you so much. And thank you again to Chiesi for supporting today's program. We really appreciate it. All right, so we are going to dive into Q&A now. And if anyone thinks of anything they want to ask in the meantime, again, feel free to put those questions in the Q&A box. All right, Dr. Kohn, first question. Can you please elaborate what you mean by evaluating, quote, performance status as shown on one of your slides?
Sure. There are these scales called Karnofsky Performance Scale or the Lansky Performance Scale, in which the doctor or nurse just rates the patient's sort of quality of activity. So 100% means doing everything. 90 might be slightly impaired in doing daily activities, and it goes down from there. So it's a very rough. It's used very commonly in oncology studies just to say how the patient is doing at the time. And so it's that's scored by the physician or the nurse at the clinic visits. And this happens with all transplant patients probably behind the scene. So it's just another way to sort of assess the quality of health. The other test that was mentioned at the same time is the parent-reported outcomes. There are sort of validated surveys that ask the parents 10 or 20 or 30 questions about either the patient or their child's quality of life that we will ask the families to fill out at the time of enrollment at 1 year and 2 years. Again, just to sort of get data on the general health of the patients before and after gene therapy.
Thanks, Dr. Kohn. Next question. If the trial is able to start in January, what will be the approximate timeline for treating all of the patients?
That's a very daunting question. So the honest answer is I don't know. So the two things that will affect it is how long the batch of vector holds out. We think there's enough probably to treat all the patients we have. The other big issue is funding. We certainly don't have enough funding to transplant all those patients. And so we would need to apply for additional support to treat more patients. And then at the same time, UCLA and UCL are going to try to license this out again. And so there's a possibility that a new company will take it over. And once again, they would be in charge of how things happen. So we're going to try and treat as many as we can, as quickly as we can. But the other sort of limiting factor is how many cell products we can make per patient. So each time we make cells for a patient, it takes, like the group of people I showed you in the photo before, it takes them almost a whole month of work to do it, because it's about a week to prepare everything to make the cells, a week to actually make the cells, and another week or so to do the paperwork and the testing of the product. And so, and we have other trials going also. And so, that's why I was, whenever I give my estimates, I try and be conservative as possible so that we're surprised in the positive way. But that's why I thought, we could probably treat 3 in the next 6 months after the start of the new year.
Thank you. This is kind of a follow-up to that question. This individual asked, how many patients can you treat with the vector you have? What is the process and timeline to get more vector?
Right. So we have about 40 aliquots, 40 samples of the vector. We think certainly for infants, one would be enough per patient. So in part depends on how many cells you have, but somewhat depends on how big the patient is, how much vector, if we need to use 1 or 2 aliquots of the vector per patient product. And that's using the newer methods to sort of use less vector per patient. And so in theory, we might be able to treat 25, 30 patients with the amount of vector we have. And, we also, vector being a biological material, it can, lose its potency. And so every year we test it and make sure it's still good. If it gets bad in a year or two, we may have to stop at that point using it. Getting a new batch of vector is about $500,000 and about a 1-year wait. So if we were to try and get another batch of vector made in Indiana, so it would be exactly like what we've used before, we'd have to make a reservation, and their waiting list is about a year. And so sort of my rough timeline is, once we're up and going and we see where we're going, if we think that we'll still be able to treat more patients, to put in another grant, ideally to CIRM, to get more data to support the FDA application. And in that budget, we would request money to get another batch of vector, and ideally would have that by the time we finish the current one. So it's sort of a rough timeline, but we think that vector should not be limiting in how many we can treat. It's more how long we control it, and if there's a new partner, whether they would want us to stop treating or not.
Thank you, Dr. Kohn. All right, next question. Again, kind of a follow-up. If you successfully treat 3 to 6 patients, what is the next step after that?
Yeah, so we kind of just touched on that. It's to try and keep treating more. So I want to treat as many as we can under this compassionate protocol, and again, with the limitations that I mentioned. And so I don't know if we will get through 26 patients. That may take 2, 3, 4 years, 2, 3, 4 years, and more vector and more funding. But we'll try.
Thank you. All right, next question. How can we stay updated on the start time to ensure things stay on track with FDA and IRB approval for a January 2023 start?
That's a good question. I'm not sure what's the best way to sort of communicate this. I don't do social media, sorry. But we do have one of the mothers of one of the patients we treated a number of years ago, who's on our committee, on our working group. So we have meetings every other week on this project to keep on track. And I can ask her if she'd be willing to to post stuff as to keep track of things. We want to be as transparent as possible, and our little feet are paddling as hard as they can under the water, but it's not always apparent, and so it's good to have open communication.
Certainly, thank you. All right, next question. What are your thoughts on improving the process to develop gene therapies like this so that they can get to the patients sooner? For example, ways to expedite FDA approvals, and your thoughts on moving therapies from clinical trials to being available on a clinical basis.
Yeah, that's a really huge and important question. And there's 2 parts to it. So one is getting sort of through the trials quicker, and one is sort of after it's approved, how is it made available? And so for the trials, I mean, FDA has gotten more flexible, and I think is continuing to, for rare diseases, not requiring a phase 1 and a phase 2 and a randomized phase 3, but to have trials like the size that we're talking about, which for many drugs is very small, to get approval based on these numbers of patients, 10, 20 patients or something. And so I think, if the trials are designed right and done well, you can get through a trial of 10, 20 patients in 2 or 3 years and possibly be applying to FDA, which still makes it sort of 4 to 5 years from start to finish. The other question that was touched on is, as happened with our ADA, Orchard has also announced that 2 other immune deficiencies that they had developed for gene therapy that came out of TGET in Milan, for Wiskott-Aldrich syndrome and for CGD. They also decided they were going to drop those projects and return those licenses back to TGET San Rafael in Milan for them to continue. And so it really, it raises questions about this model of commercialization of these expensive therapies for rare diseases. And, it's all been sort of supported by venture capital with the belief that this will be profitable and give them a return on investment. And, it's complicated making these products. I showed you our little ragtag group of, 8 people doing these trials. A company, it's going to take them 100 people to do this, because the regulatory burden, the facility requirements, so many of the things to do commercial manufacturing are so much harder than what we do at an academic standard, that's where you hear these drugs are costing $1 and $2 million. It's not, there may be some profit in there, but it's also, their costs are so much higher than when we do it in our university building. And so there's an effort to see, is there a model separate from commercialization? And so in fact, next week in London, Claire Booth has been leading an effort in Europe with representatives from multiple countries across the EU and the EMA, the European equivalent of FDA, participating to see, can we develop a model where these would be, for example, the ADA would continue to be made at UCLA, although it's not licensed or it's not marketed, we can get reimbursement for what it costs so we can sustain the enterprise. And there's very little precedent for that. And the manufacturing standards, as I said, for approved drugs are probably higher than a university can go to. But I think that's one of the things for all rare diseases, not just immune deficiencies. We need to look at a way to make these therapies available at a cost that can be afforded. And ultimately, in the U.S. at least, it's going to come down to individual insurance companies agreeing to pay for them if FDA allows that model to occur. And we think besides the benefit to patients, it's also cost effective. And to have an autologous transplant with gene therapy costs less than an unrelated donor transplant. It costs less than a lifetime of enzyme therapy. And so for countries with national health, I think that savings could be more appreciated, that you put out money this year, but you save it over the next 10. In the US, with people changing insurances and all that, it's a little harder for the benefits to be appreciated, but hopefully we can get to some way that these can be made available because, so the benefit gets to patients. Kind of a long answer, but it's something that there's a lot of discussion. And there's several efforts in the US. Jennifer Doudna, who got the Nobel Prize for describing CRISPR at UC Berkeley, has an institute. And one of their main goals is to look at how to make the CRISPR therapies available affordably and widely. And there's so there's several efforts trying to address this problem, that now we have these therapies after several decades. How do we make them available?
Wonderful, thank you. All right, next question. Dr. Kohn, how many patients are required for a successful clinical trial and FDA reapproval? Do you plan to recruit ADA-SCID patients from Canada? And if so, do you need Health Canada's permission for this?
Yes, so there's no fixed number. And you basically have to have the number that gives a data set that's convincing. And so some of it will be the statistics of it. So if for example, you're going to compare the survival with gene therapy compared to survival with standard transplant, you need to have enough numbers in both groups to be able to make a statistically valid conclusion that it's better or it's equivalent or something. I showed you the PIDC transplant data. Even with those large numbers, that 10% difference wasn't big enough numbers to show statistical validity. I think that's the model we want to get away from, and just show that it's beneficial and get approval. So the number is not fixed. And Orchard thought that the 30 patients that we had would have been sufficient to get FDA, and seemed to have been given that indication from discussions with FDA. What was it? The last part was?
They wanted to know if you'll be recruiting patients from Canada and if you need the permission of Health Canada to do so.
Yes, and so yes, our list includes U.S. and Canadian patients sort of in the order of referral. And so yes, and so the way it worked previously was sort of on a province by province basis. I don't understand the Canadian health system deeply, but my understanding is that sort of each provincial government to some extent regulates their costs. And so if we want a patient from Ontario province to have gene therapy, the doctors there have to make a request to the Ontario Health Ministry for them to pay for the cost. And in the past they have, because again, the savings over a few years, if they don't need enzyme therapy, makes it cost effective for them. And so I think we were able to get approval for all the Canadian patients to pay for the hospital costs. The gene therapy part, the vector, the cell modification, the lab tests come from the grant. But that's because that's unpredictable. That's why we can't be specific on how many patients we'll treat. The more we can get their home insurer, their home government to pay for the clinical costs of the transplant, the more we'll be able to treat.
Thanks, Dr. Kohn. This next question, someone emailed in, so forgive me while I read from my phone. You said there are 26 people on the list, but not all have been contacted. Does that mean that some patients placed in the line of 26 individuals may improve come 2023?
Possibly, although I can't say. So, you know, as I said, it seems a little early to start contacting people and letting them know that they're 18th in the list or something. But it is possible that if some of the patients have had a bone marrow transplant or something, that people may move up in the order.
Thank you. All right, next question. Just to clarify, are we saying that all of the ADA-SCID children on the list will be treated, or do we still need funding to have them in the trial?
Yeah, so we can't guarantee that all will be treated. And you know, I talked about the factors about vector, money, time that could influence it, and so we'll try to treat as many as we can. But it's not a guarantee if you're on the list that it will be available.
Thanks, Dr. Kohn. And thank you so much to our audience for all this engagement and these wonderful questions. It definitely makes it for a more robust lunch and learn, so thank you. All right, this last question is kind of looking ahead to the future of gene therapy. Can you explain about, and you might not be able to answer this, can you explain about gene therapy for Duchenne muscular dystrophy?
Sure. So Duchenne's, I think it's pronounced, is one of the more common and more severe forms of muscular dystrophy. So it's also like ADA-SCID, a single gene defect. It's a gene for a protein dystrophin that's involved in muscle production. And so the basic idea is replace that gene like we're doing for ADA. It's much, much more challenging because you have to deliver the gene to the muscles in the body. You can't take them out and put them in a cell culture dish like we can with bone marrow stem cells. So it's all about delivery and delivering the gene to the whole skeletal muscle system, to the heart, to the diaphragm. And so there are early trials starting using often adeno-associated virus as a different vector injected into the bloodstream. And ideally it goes to the muscles and introduces the gene. And so it's a challenging, it's more challenging, I think, than even, you know, what we've talked about, the blood cell diseases. But there's progress. There's also other delivery methods that are being looked at, like the way we get the COVID vaccine in RNA in sort of a fat globule, at that as a way of delivering. And so it's moving forward. I believe there are some early trials underway looking at using viral delivery in the body for AAV, for Duchenne's muscular dystrophy.
Wonderful. Well, thank you so much, Dr. Kohn, for addressing all those questions. And if anyone else thinks of anything they'd like to ask, they can always reach out to IDF and ask a question to IDF. So if you have any more questions, feel free to reach out to us at primaryimmune.org/ask-IDF or give us a call at the number on your screen. Thank you so much again to Dr. Kohn for his time and expertise today. We just have a couple of quick slides we're going to close out today's session with. So IDF has a ton of resources on gene therapy, both for SCID and other forms of PI, and we wanted to just share a couple of helpful links to the clinicaltrials.gov, as I'm sure everyone is familiar with. Also to IDF's gene therapy overview. We do try to keep our gene therapy pages as updated as possible. And we also have some great Q&As with other leaders in gene therapy, as well as interviews with families who have gone through this treatment journey. So we definitely encourage you to check those out. We want to close by saying that all IDF programming is truly guided by the needs of the PI community, and we want to hear your feedback. So please take our quick survey that will be emailed out to everyone after the program and let us know if you like today's presentation. Finally, we hope to see you at our next IDF Lunch and Learn, which is taking place in 2 weeks on September 28th, where Rebecca Marsh from Cincinnati Children's Hospital will provide us with an overview of HLH. So that wraps it up for today. Thank you so much again, Dr. Kohn, for your wonderful presentation and for sharing all this really important information with the SCID community. Thank you, Andre, for your word from Chiesi. We definitely appreciate you supporting today's program and hearing from you. And thank you everyone for your participation and engagement in today's Lunch and Learn. We hope to see you at the next IDF event. Thank you again and have a great rest of your day. Take care.
Thank you, Emma.